Assalamualaikum warahmatullahi wabarakatuh. My name is Tania Askia from Biology 2017, State University of Surabaya. I would like to present about cell division with model direct instruction. Assalamualaikum warahmatullahi wabarakatuh. How are you? All of you are good, right? Alhamdulillah. Before we start, let's pray together. Finish. Okay, today we will discuss about cell cycle and cell division. The chapter outline that will discuss about importance of cell division, cell cycle, regulation of the cell cycle, cell division, prokaryotic and eukaryotic cell division. Why can living things grow big? Sure, there is a process occur in their body. So let's watch this video to know that.
Hey, this is Coach Boyson, and in this screencast, we're going to be looking at the importance of cell division. So why is it important that cells divide? Well, you can see the two on the screen here. One of them is growth. Cells divide, allowing us to get bigger. That's why we've gone from one cell when we were conceived to trillions of cells today. Also repair. When cells are damaged, we've got to have a way to replace them, and one way we do that is by cells replicating to replace those damaged cells in the first place. And so when we think about cell division and its importance, I want you to understand that we are not one cell that just got bigger. We are actually one cell that replicated into 2, which replicated into 4, which replicated into 8, so on and so on. To again, we are made up of trillions of cells today. And so we went from one cell to trillions of cells. What's really unique is all of these cells carry the same DNA. And so cell division, especially with mitosis and body cells that we're mainly looking at today, is going to occur and produce cells that are genetically identical, have the same DNA. And so all of our cells have that because they've all replicated from an original cell. And so to understand these concepts that we're going to be looking at on cell division in this unit, we have to understand this idea of haploid and diploid. So if we look at our first one, haploid, you'll notice it just has one set of chromosomes. That's why we always mark it with just N, as opposed to diploid cells, which are going to have 2N or have 2 sets of chromosomes. And so our haploid cells that we find in our body, that is going to be our sex cells. And so in this case, that is our sperm and our egg. Now humans in our body cells, our somatic cells they're called, our body cells have 46 chromosomes. Well, if our body cells have 46 chromosomes, sex cells are haploid. They're different. They only have half that number. So a sperm is going to have 23 chromosomes, an egg is going to have 23 chromosomes. So sex cells are always going to be haploid, and the way I remember that, haploid starts with ha, kind of sounds like half. So sex cells are always going to have half the number of the body chromosomes. And so in this case, the diploid cells, which are our body cells, like I said, sometimes called somatic, they're actually going to have 46 chromosomes, or 2 sets of chromosomes. And so they're called diploid. And so what happens is when this sperm and egg join together and fertilize, what we get is we get a zygote. And this zygote is going to be a combination of this sperm and this egg. And so when you add 23 from the sperm, 23 from the egg, that new zygote now has 46, which is what organisms like us, humans, have. That's how many chromosomes our body cells have. Now, every organism is a little unique. So like if I told you the body cells of an organism had 100 chromosomes, well, that would mean the sex cells would have to have half. Why? Because the sex cells are haploid. They always have half the number of the body. So if the body had 100 chromosomes in the body cells, for that organism, the sex cells would carry 50. And so in this case, ours has 46, so the haploid cells, sperm and egg, are going to have 23. And so diploid is our body cells, 2 sets of chromosomes, haploid, just half. And so if we look at this process, we know the first cell that makes us who we are is that zygote. It has our DNA, it has 46 chromosomes, and then it's going to begin to replicate. And if we look at this process, you can see day 1, there's our zygote. By day 2, it's actually 2 cells. It's divided one time. It's divided again by roughly midday, and then divided even again by day 3. And so it's about 8 cells. And then by day 4, those 8 have continued to replicate and replicate. Day 5, this is called a blastocyst. That's about 150, 130 cells. And then day 5 also, what happens, you probably didn't know this, I didn't know it until I started studying it, we actually hatch from an egg. So our little embryo here, our blastocyst, actually will hatch from its egg to implant in the female's uterus, in the side wall of the uterus, to begin developing a connection with the mother there. And so kind of interesting there. And obviously this little situation here is very close to me. Me and my wife had trouble getting pregnant, so we had to do what they call in vitro fertilization. They took her egg, like you see up here at the top left, and in a petri dish down here, down at the bottom, they actually injected one of my sperm directly into the egg and fertilized it directly. It grew in a petri dish for 5 days, and after 5 days, if you look down here, I actually have a picture of what my son looks like at 5 days. Most people probably don't have that. This is the blastocyst stage. He's about 150 cells or so, and you can see this little mass right here. That is the portion that is going to become him as these cells continue to divide and replicate. And so why is cell division important? Again, it's how we grow. Again, we said we can also repair from it, but it's how we grow. And so in this picture here, this is him after about 7 weeks. You can see him sucking on his thumb there. This is 12 weeks. He's even bigger. He has his fingers. Everything's going on. This is 22 weeks. Getting pretty close there to coming out. And then today at 3 years of age. And so he's just going to keep getting bigger and bigger as it goes because his cells are dividing. And obviously at his age, with the growth rate that he's going through, his cells are dividing at a much greater rate than, say, somebody my age are replicating that. And so, again, why is cell division so important? Well, it's what allows us to grow as organisms. It's also what allows us to repair damaged cells. And so when there is damage, such as a cut, we can grow and heal and repair that because of cell division. So hopefully that was helpful. I'm Coach Boyston. You guys have a good day.
From the video, we know that cell division can cause living things grow big. The importance of cell division are for growth, development, repair of worn-out tissues, and reproduction, to facilitate the exchange of materials, and to control DNA overloading. So, after our lesson, the learners will be able to explain the cell cycle, identify the characteristics and stages of mitosis through microscopic observation, identify the sequence of stages of meiosis, identify the characteristics of the stages in meiosis, differentiate the process, stages, place of occurrence, the function of mitosis and meiosis division. And last, make onion root incision preparation to be observed microscopically. Observe the union root incision sites using a microscope and present data from cell division analysis through written reports. Next, for the method, first, the cell cycle is the sequence of events from the time a cell first arises as a result of cell division until the time where the cell itself divides. This consists of periods of growth and development, DNA replication, preparation for division, and cell division. Cell after division begins a new cycle. The cycle consists of two main periods that are interphase and mitotic phase. Then, cell division is the process by which a cell divided to form two new cells. There are three types of cell division or cell reproduction in organisms. Number one, prokaryotes or bacteria that occur binary fission that divides, forming two new cells that cell identical. Number two, eukaryotes. There occur mitosis and meiosis. Eukaryotic cell division that result in two daughter cells, each having the same number and kind of chromosomes as the parent cell. Number one, mitosis. There are two main steps. Number one, prophase, metaphase, anaphase, and telophase. And then cytokinesis that occur, cytoplasm divides, forming two new daughter cells. Each daughter cell is genetically identical to parent cell. For more information about mitosis and cell cycle, you can look at the
The human contains an estimated 100 trillion cells, and yet we start life as a single cell. To grow, develop, and repair tissue damage, we rely on cell division. In eukaryotic cells, this process is accomplished by a series of well-orchestrated steps called mitosis. Every day our bodies must produce millions of skin cells to replace those lost through normal activity. Each of these cells must have a complete complement of the genetic material. Prior to cell division, where the cell divides into two identical cells, the DNA needs to be replicated so that each daughter cell receives an exact copy. Following DNA replication, the chromosomes condense in the nucleus of the cell. DNA condenses by wrapping around cores of histone proteins, forming nucleosomes. This beads on a string structure is called chromatin. As a cell prepares to divide, chromatin coils up further, shortening and condensing the chromosome. The replicated chromosomes are called sister chromatids. The replication of DNA and the formation of sister chromatids is one part of the entire cell cycle. To prepare for cell division, the cell goes through interphase, which can be divided into three distinct phases. During G1 or gap1 phase, all the organelles and cytoplasmic components, including the centrioles in animal cells, replicate. Then during S or synthesis phase, the DNA replicates. Finally, during G2 or gap2 phase, all the enzymes needed to aid in the process of cell division are produced. Most eukaryotic cells spend a great deal of time in interphase and a very short period of time actually dividing, a process called mitosis. The cell is now ready to go through mitosis, which consists of prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes condense and become visible, appearing as two sister chromatids held together at the centromere. The cytoskeleton disassembles as the spindle begins to form. In animal cells, centrioles play an important role in the distribution of the chromosomes in the dividing cell. The centrioles migrate to opposite poles, establishing a bridge of microtubules called the spindle apparatus, and the nuclear envelope breaks down. Towards the end of prophase, chromosomes attach, by proteins in their centromeres called kinetochores, to microtubules from each pole, moving the chromosomes toward the equator of the cell. During metaphase, all chromosomes are aligned at the equator of the cell, called the metaphase plate. Anaphase begins with the degradation of proteins that hold sister chromatids together, freeing individual chromosomes. The free chromosomes are then pulled by their kinetochores to opposite poles. At telophase, a cleavage furrow forms in the center of the cell. This indentation is made from a constricting belt of actin filaments surrounding the inside of the cell's circumference. Chromosomes cluster at opposite poles and begin decondensing as the nuclear envelope reforms around them. The spindle apparatus disassembles as the microtubules are broken down into tubulin monomers that can be used to form the cytoskeleton of the daughter cells. In animal cells, cytokinesis completes cell division by extending the cleavage furrow to completely separate the newly formed daughter cells. Since plant cell walls cannot be constricted by actin fibers, vesicles form an expanding membrane partition called the cell plate. Like animal cells, plant cells use cytokinesis to finish the division of the contents of the cytoplasm between the two identical daughter cells. During the cell cycle, certain checkpoints are encountered to make sure the process is occurring accurately. And if it is not, the cell cycle will stop at the checkpoint and correct, or possibly inhibit that cell from dividing. The first checkpoint is the G1S checkpoint, and is considered the primary point at which the cell cycle continues or stops. External signals and growth factors can influence the cell cycle and affect the progress at or before this critical checkpoint. The G2M checkpoint allows cells that have successfully completed all 3 phases of interphase to begin mitosis. The last checkpoint is the spindle checkpoint, ensuring that all chromosomes have attached to the spindle in preparation for anaphase. Growth factors, the size of the cell, and the nutritional state of the cell are all contributing factors in cell cycle regulation, ensuring that only certain cells divide at appropriate times. Once all the checkpoints in interphase are cleared, mitosis can occur. From interphase to cytokinesis, the entire process of cell division can take on average 10 to 20 hours in a typical plant or animal cell. Depending on the nature and use of the cell, the process can happen at different frequencies as well. In humans, our skin cells have a high turnover rate due to wear and tear and go through mitosis very frequently, whereas other cells, such as adult neurons and muscle cells, rarely divide. The accuracy of mitosis, as well as the consistency of the checkpoints during interphase, ensure that most cells in a eukaryotic organism can produce identical copies of themselves. This process allows for growth and repair to prolong overall physiology as well as life itself.
Number 2 is meiosis. In meiosis, same with mitosis, but in meiosis, cytokinesis, each daughter cell is not genetically identical to parent cell. For more information about meiosis, you can look at the video.
Have you ever wondered why children do not look exactly like their parents, or why some siblings look so different from each other? The answer lies in the process of meiosis, which generates the gametes for reproduction. Meiosis is the process by which haploid cells are produced from a diploid cell. For this to occur, the chromosomes must be correctly sorted and distributed in a manner to create genetically unique cells with half the number of chromosomes as the original cell. Meiosis occurs in special cells called germ cells within the gonads of males and females. For the successful reduction in the number of chromosomes to occur in the new haploid daughter cells, 2 rounds of divisions are necessary. These divisions are termed meiosis I and meiosis II. Although the processes of mitosis and meiosis look similar, there are a few major differences. One obvious difference is that mitosis results in 2 diploid daughter cells, and meiosis results in 4 haploid cells. As in mitosis, meiosis begins after a cell has successfully completed the G1, S, and G2 stages of interphase. During the S phase of interphase, the DNA is replicated, producing 2 copies of each chromosome called sister chromatids. The paired centrioles in the cytoplasm duplicate and begin extending microtubules that will form the meiotic spindle. Sister chromatids remain attached at the centromere and condense as the cell enters prophase I of meiosis. Up to this point, the cell looks similar to mitosis, but 2 events occur in meiosis that do not occur in mitosis and lead to genetic diversity. The first event occurs during prophase I, when homologous pairs of sister chromatids lie side by side in a process called synapsis, forming a tetrad or bivalent. The homologous chromosomes share similar but not necessarily identical genes. Once this structure is formed, the second event, called crossing over, can occur. During crossing over, a physical exchange between chromosome segments of non-sister chromatids occurs, increasing genetic diversity. Prophase I concludes with the fragmentation of the nuclear envelope as the duplicated centriole pairs move to opposite poles of the cell. As they move, the centrioles extend spindle fibers forming the meiotic spindle. In prometaphase I, with the paired centrioles in place, the meiotic spindle is fully formed. The sister chromatids attach to the spindle fibers by their kinetochores. Here, another key difference between mitosis and meiosis occurs. Because of synapsis and crossing over in meiosis, homologous chromosomes remain aligned, so that a pair of sister chromatids is attached to only one pole by the kinetochore microtubules. In mitosis, each sister chromatid is attached to a spindle fiber. The fibers originate from opposite poles. During metaphase 1, bivalents randomly align along the metaphase plate. Due to independent assortment, this alignment is random and adds to genetic diversity. In anaphase 1, the homologous chromosomes separate and move toward opposite poles. Meiosis 1 ends with telophase 1, when the chromosomes decondense and the nuclear envelope reforms. Cytokinesis separates the cytoplasmic material, and the two daughter cells are separated by a cleavage furrow. Since the final product of meiosis 1 is two haploid cells, meiosis 2 begins without the chromosomes going through another round of DNA replication. Centrioles again duplicate and begin moving to opposite poles of each cell. In prophase 2, the sister chromatids condense while the spindle starts to form as the nuclear envelope disappears. In prometaphase 2, the sister chromatids attach to the spindle by kinetochore microtubules, with sister chromatids attached to opposite poles. The spindle aligns the sister chromatids along the metaphase plate during metaphase 2. During anaphase 2, sister chromatids separate and individual chromosomes move toward the poles. The entire process ends with telophase 2, as the chromosomes decondense and the nuclear envelope reforms. Cytokinesis occurs and cleavage furrows separate the two daughter cells into 4 haploid daughter cells. The haploid daughter cells will specialize into gametes, either sperm or egg. These fuse in fertilization to form a zygote, which will grow into a child. The child receives half its chromosomes from its mother and half from its father. Because men and women produce millions of gametes, and the selection of gametes in fertilization is random, this contributes to genetic diversity. This explains why a child isn't identical to either parent. But why aren't siblings identical? The diversity comes from several sources. In prophase 1, non-sister chromatids can exchange DNA through crossing over, increasing the genetic diversity of individual chromatids. In addition to crossing over, in metaphase 1, the pairs of homologous chromosomes align independently along the metaphase plate and sort independently into the daughter cells. This process, called independent assortment, produces 4 genetically distinct haploid gametes. As the total number of chromosomes increases in an organism, the number of genetically distinct gametes increases by 2 to the nth power. This means an organism with n equals 3 can produce 8 unique gametes. For humans where n equals 23, there are 2 to the 23rd power unique gametes formed, or over 1 million different possible combinations. The combination of independent assortment, crossing over, and the random pairing of gametes during sexual reproduction increases genetic diversity and explains why a child will not look exactly like his or her parents or siblings.
After that, you must make a group with 3 students for each group, and I will demonstrate about our practicum. Then, you must do your practicum independently and do your worksheet. Okay, for the first step, you must cut the onion root 5 mm using a razor blade. And then, soak the onion root in a solution of alcohol and distilled water until 4 minutes. After 2 minutes, suction alcohol water on a petri dish using paper tissue until dry. And then drop the onion root with 2 drops of HCl and 2 drops of water for 2 minutes. And drop of 2 water. That is. And then suction of HCl and water in petri dish with paper tissue until dry. And then cut the onion root and place it in the object glass. You must be careful because it will be thick. And then drop the onion root with a drop of methylene blue. Then cover the onion root with a cover glass until the preparation is flat. And then preheat the preparation using a Bunsen burner and don't boil it. Just preheat. And then observe in a microscope and see what happened. Now, you must doing our practicum independently and do your worksheet. Do you need some help? Oh, this is the bed slide. This is look so thick. You cannot see what happened in the microscope. And this is good slide. This is so thin, you can see what happened in the microscope. So you are doing now, see what happened. After you do your experiment, you know that cell division always occurs in living things that cause for your growth and repair your body cells. From your experiment, we can conclude there are 4 steps in mitosis that are prophase, metaphase, anaphase, and telophase. In the prophase, the chromatin strands in the nucleus thicken, shorten, and form chromosomes. In the metaphase, chromosomes are in the equatorial plane. In the anaphase, chromosomes become 2 parts so that the chromatids were in opposite direction. In the telophase, 2 nucleoli are formed in the cell. After doing your experiment, you must assign next week, and I think enough. Before we close, let's pray together. Finish. Alhamdulillah. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.